I welcome you all again to the second session of day two. For this, we have our esteemed speaker, Professor Uzair Alam from Jamia Hamdard. And before Sir actually starts, I request Professor Naseemul Huda Sir to kindly felicitate Sir by offering floral. Uh, by offering a sapling and a shawl uh, as a token of respect. हाँ ये फोटोग्राफर हैं हमारे शॉल देना इसको मोमेंटो टॉप के बाद दे देते हैं So, I take this opportunity to introduce Professor Uzair Alam. Sir is currently working as a professor from School of Pharmaceutical Education and Research from Jamia Hamdard, New Delhi. And as all of us are aware, Jamia Hamdard it is a leading institution with NIRF rank 1 in pharmacy, and also it has a remarkable ranking in World QS ranking, that is 94 in the field of pharmacy and pharmacology. Professor Uzar's leading authority is in the field of pharmaceutical chemistry, and his group heads in medicinal chemistry and molecular modeling lab, where his groundbreaking research in drug discovery has garnered international acclaim. Sir was also selected as top 2% scientist by the list issued by Stanford University, USA in the year 2024. Professor Alam's primary focus is on combating diabetes and cancer. He is actively involved in computer aided drug discovery and molecular modeling methods, which find application in cancer treatment. And recognizing his outstanding contributions to the field, Sir has been awarded by various prestigious awards, which includes Young Scientist from Department of Science and Technology DST and SERB, that is Science and Engineering Research Board. His research is supported from grants both by ICMR and DST SERB, which has significantly advanced our understanding of these critical areas. Under Sir's guidance, there are 10 PhD research scholars who are currently engaged in current cutting edge projects that he directly supervises. And additionally, his academic career boasts over 1,000 academic research articles published in peer-reviewed journal with over 4,000 citations and a remarkable H index of around 40. With these words, I invite Professor Uzair to please continue with his talk entitled Biological Applications of Oxygen and Nitrogen Containing Heterocyclic Analog.
Uh, Thank you, ma'am, for the nice introductions. Good morning to everyone. So, today's title of this GAN programs is Biological Applications of Oxygen and Nitrogen Containing Heterocyclic Analogs. So, I am not going to here, giving the detail about what kind of activity I am going to perform by cell line, in vitro assay, enzyme assay, or something. I'm here just going to discuss about how the because we have synthesized a lot of compounds, but until unless if you are not going to get the output in terms of bioactivity. So the docking is one of the tools, or dynamics, molecular docking, or in silico approach, one of the tools by which we are going to predict the activity of the design molecule, so that we can save our time in lab, or we can save our money as well as so that's why we just have to synthesize a lot of compound in the lab. If suppose we can get the 5 hits, 1 hit, so we can perform all the in vitro, in vivo, cell line, everything we can get performed. So that's why here I'm going to talk today in terms of molecular dockings. And so how many of you have aware of this molecular docking? I think it's not new for all. Uh, so, here, so in protein ligand docking, basically I'm working in Schrodinger software since 2013 when I got PhD Young Scientist, and that I've just purchased that software. So at that time, what I thought, what should I purchase so that I can enhance my knowledge? Because I saw at that time the research paper which were published from the south or anywhere in Elsevier on that thing. So I thought that whether what is these things is docking. In paper these images looks very good. So whether I am able to learn it or not. So for that, I can devote, 2 to 3 years to learn all these techniques of that molecular dockings and dynamics by this software. So I'm just giving the few of these slides, a few of my research and few of my case studies in this presentation so that we can learn. And one more thing is that I can promise you within 2 or 3 days, I send one of my research scholar who is just going to, workshop, yeah, hands-on for 3 to 4 hours on Schrodinger software, where you can easily learn molecular dockings and dynamics, pharmacophore modeling, virtual screening, so all these techniques. So within, I just have to give my words to Professor Raisuddin Saab. So in protein ligand docking, what we have to see, there's the two things in docking. One is called as the search algorithm. So here in search algorithm, we have to generate the large number of poses by the ligand in the binding site. Suppose if I am taking one molecule, so that molecule have so many chiral center, they have the different orientation. If somebody who is sitting in that chair, so sometime what happen, I'm having sitting in this way, that way, in what pose I'm just getting. So I just have the different way to sit in the chair. The chair is the same, okay, but the way I'm just sitting, it may be the different from other. So that's why in case of search algorithm, we have to generate large number of, ये इसमें लगाना पड़ेगा रईस भाई ये चीज ठीक ये देखिए कौन सा किधर है बताइए हाँ वो अच्छा 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 ये पॉइंटर है ना अच्छा इस पे पॉइंटर नहीं आ रहा ना
पॉइंटर सर इस पे नहीं है
नहीं आता है ना
वो आएगा जी नहीं आएगा
ओ
सर वो
अच्छा 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 नहीं आएगा ठीक ठीक
ठीक नहीं वो उसका जो इंटरप्ले
अच्छा अच्छा ठीक ठीक so here I am taking one of the compound from chicory plant. So here this is the structures of chicory acid. So as I see, this chicory acid have dihedral angle, torsional angle. So they have to change their, shape or orientation by change their dihedral angle, by change their, torsional angle. So they have to generate the large number of, conformers. So you can see here, I'm just taking 9 of these most stable conformer. And these conformer are just going to be, these were the ligands which were going to bind with that of the protein. So this is the target site of the protein, or you can say the binding type site. So in docking, there is an interaction of two molecules. One is called as the small molecule, another is called as the large molecule, yeah, macromolecule, that is called as the protein. That is called as the protein or receptor. So here you can see the chicory acid, they have to form a different shape by changing their dihedral angle, by changing their torsional angle. So here this is the binding site, and this may be one conformer, may this be this way, that way. So there are different poses which actually going to bind on the active sites of the binding sites of the. So here I am taking this is the protein of the alpha glucosidase. Alpha glucosidase is the one of the major target for the development of anti-diabetic agents. So there are a lot of anti-diabetic targets. What? SGLT2, DPP4, GLP1 agonist, PPR gamma. So alpha amylase, alpha glucosidase. So here I am taking the large, protein that is called as the alpha glucosidase. So here we just going to put the ligand into the binding sites of the alpha amylase for the. So now, the first thing we have to see the search algorithm. The first thing is that search algorithm, and second thing is that scoring function. So what do you mean by scoring function? Because once the ligand interact with the receptor, they may just have, they have to form certain type of interaction that is called as the non-covalent interaction. It may be the hydrogen bonding, it may be the van der Waals interaction, it may be the pi-pi interaction, it may be the pi-cation interaction. So here basically in docking we have to find out the binding affinity of particular ligand for the particular target site or protein or the receptor. So how we have to say that our ligand molecule have less affinity or high affinity? So for that we must have some scoring function or some energy in the form of kilocalorie per mole. So here in the binding affinity is sum of the van der Waals force energy, electrostatic, hydrophobic and hydrogen bonding. So basically here we have to calculate the interaction energy between the ligand and the receptor. If your ligand is more stable at the binding site, so that means which we have. So scoring function basically we have taken in form of negative. So suppose one ligand have the two conformer, one having the minus 10 kilocalorie per mole, another having the minus 8 kilocalorie mole. Which one is the high energy? Which score we have taken for? Minus 10 or minus 8? Minus 10, because minus 10 is the less energy, and less energy means more stable at the binding site. So here, and one more thing is that in Schrödinger there are the three way by which we have to perform the molecular docking. One is called as the HTVS, high throughput virtual screening. Second is called as the standard precision, and X is called as the extra precision. So suppose if you have a 1 lakh millions of the molecule, so you are not going to perform the extra precision docking, because here you need to have to run one ligand molecule for docking. It required 2 to 3 minutes. So it's not possible to dock the millions of compound through the virtual screening. So for that initially we have to get through the HTVS. But one more thing is that one, you have to get the large number, and you use the speed is high but accuracy is less. So accuracy is more in case of extra precisional. So once you have to take in 10,000 molecule, after HTVS you got the 1000, and after 1000 through the SP you got the 50, and 50 you go for the extra precision, and finally you get the hit 2 or 3 molecule which have the good score or good binding. So here in docking, so here in molecular docking or ligand protein docking, two things is there. One is called as the search algorithm, and the second is called as the scoring function. In case of search algorithm, what we see, we have to find the different conformers of, we have to generate the different conformers from the ligand by changing their dihedral angle or torsional angle. And each of these ligand, each of these conformer will have differently bind with that of their target site or the protein. So in docking there are so many docking. One is called as the rigid docking, another is called as the flexible docking. Our protein is rigid or flexible? It is a rigid structure. But ligand may be the flexible or may be the rigid. How you have to introduce the rigidity? By incorporating certain double bond or something which are not going to rotate in their 3D space. That is called the rigidity. So most of our docking we can perform by the flexible docking. So in case of flexible docking, your receptor is, sorry, rigid docking, your receptor is rigid and ligand may be the flexible or maybe the rigid. So on that. But there are some way, if you perform the rigid docking by some molecule, but you are not going to get the good output or enough output for activity. So in that case, you have to perform the induced fit docking. Your ligand is not properly going to interact with the receptor. In that case, what? You have to change the receptor orientation so that the molecule is going to interact the particular site. So here in case of induced fit docking, what we see, suppose I am taking hypothetically one molecule which having like catechol type of molecule. Here they have to make a van der Waals hydrogen bonding and here they have to make a ionic interaction. So this is the receptor, this is the binding site and maybe one of the amino acid lysine or they have the free so C terminal which having the CO minus charge. Here this catecholamine, once it get accepted proton, it may be protonated charge. So they have to make a ionic interaction, they have to make a hydrogen bond, they have to make a van der Waals interaction. But the thing is that these molecules bind with the receptor by 3-point attachment, but 2 is very strong and 1 ionic interaction is very weak. So in that case, what happened? Receptor itself change their shape. Receptor itself change their shape so that the anionic carboxylate group get closer to that of the cationic amine, so that they have to form a strong interaction. So this is called as the induced fit. Induced fit is the term by which the receptor itself change their shape to accommodate the ligand for better interaction. This is called as the induced fit docking. Another docking they are called as the covalent docking. Suppose I am taking example of beta lactam like penicillin, ampicillin, amoxicillin, whatever they. So penicillin, so here there is a site that is called as the penicillin binding protein site. They have to make a ionic interaction with the lysine. They have to make a, here what happened? This pi bonds move here, this carbon become delta positive, this carbon become delta. So there is a one enzyme that is called as the transpeptidase. So transpeptidase are helped to make a cross linking with the NAM and NAG so that the cell wall get grow and the bacteria will grow. So in transpeptidase, what happened? There is a serine residue, that is the amino acid, and serine having the free hydroxyl group, that is OH, and OH oxygen having the lone pair of electron which act as a nucleophile, and that nucleophile can attack on the electrophilic carbon. How these carbon become electrophilic? By moving the pi bond here. This is the delta negative, this is the delta positive. So once it get interact serine, so they are covalent. That means they are never detached from the, so this is called as the irreversible binding. In case of reversible, they are the short one. Once the activity get achieved and after that the receptor get released from the ligand, released from the receptor binding site. But in case of irreversible, in case of covalent bonding, there is no any ligand is detached till the new receptor is synthesized within the system or within the body. So this is called as the irreversible or covalent docking. So these are the types of docking like rigid docking, induced fit docking, covalent docking. And so in Schrödinger, sometime how many of you have to search the PDB ID in Google or something? Protein data, have you seen? Any of you? Raise the hand. So you have to search the PDB ID, suppose. And sometime what happened? You are not going to find any co-crystal structures. So in that case, how you have to perform the dock? Because once you have to make remove the co-crystal from the binding site, then you are just going to impose your own ligand within the binding site, which can be occupied by your co-crystal. But if you don't have the protein which don't have the co-crystal ligand, what is the binding site you have to find out. So in Schrodinger there is a one tool that is called as the site map, or maybe some other tools in Schrodinger they are used that is called as the homology modeling. In case of homology modeling or site map, when you don't have any co-crystal ligand. So in that case, how you have to find out the binding site? Because this is the whole protein. So in whole protein, how can you say that which is the binding point? It may be here, there, for. So for that, we have to perform the site map analysis to identify the binding site on if you don't have the information about the bound ligand on the protein structure, if you don't have the information about the binding pocket residue, you can identify to top rank. If you don't have two co-crystal ligand present in the binding site, to understand the binding site properties which can be used to lead optimization, use the 3D structures of target receptor to design or search potential drug compound that can be modulate the target function. So within 2 or 3 days, if I will send one of my research scholar, he can perform the site map analysis by Schrodinger, then you can able to understand properly what actually happened. So what happened, because once we have the protein, so if your ligand is going to bind with that protein, so how, because this is the structure, this is the ligand, and this is the whole protein structure. So in protein structure, some part is called as the hydrophobic area, some part they are called as the hydrophobic region, they are called as the ligand hydrogen bond acceptor region. This is the acceptor, and this is ligand hydrogen bond donor region. This is the green one, donor region. So here this one is the yellow one is called as the hydrophobic region. So if you have to find a protein where these hydrogen acceptor, donor, hydrophobic area, so in that case what happened, the ligand must have the, here is the acceptor, so ligand must have the donor. So if you see here this is the amino acid, here this is the N and H, and here is the nitrogen, that is the pyridine ring. So pyridine is what? They are hydrogen bond donor or acceptor. They are the acceptor because they don't have the hydrogen. But here this is the amino acid which are present in the binding sites of the protein. So this one is the hydrogen bond donor. So donor bind with the acceptor. Similarly here, if you see that's the hydrophobic, this is the phenyl ring, the benzyl oxy group is there. So this benzyl group is make as an hydrophobic part. So they have to cover up this yellow part of the hydrophobic binding site. So in site map analysis, how you have to perform? Once we have to run the site map, so there are, suppose in protein, we have to identify the 5 sites, 1, 2, 3, 4, 5. So how can you say that which site is the actual binding site? So on the basis of site score, D score, volume, size. So out of these 5 sites, which having the highest site score? 1.04, which means the D score. D score is the drugability score, which is, and volume, which is the large area. So out of 5, so here this one is the site 1, this is the site 2, site 3, site 4, and site 5. So here in site map 1, this is the binding area. This is the whole protein. I have taken the EGFR kinase protein. So here this part is called as the binding site. This part is the binding site. This part is the binding site. Here this is the 4th binding site, and this one is the 5th binding site. So out of 5th binding site, so this one is the actual binding sites of the EGFR protein. So on the basis of what? Site map score. So what is the site score? So site score, they have this equation 0.0733 square, and this one, and D score is the drugability, and binding site volume is in the A angstrom square, and here N is the number of site point, and E is the enclosure score, and P is called as the hydrophilic score. So here if D score, drugability score is more than 1, which is considered to be as in case of site 1, what is the D score? It is 1 point something. So if the D score is more than 1, then we have to consider. If the value is less than 0.6 or 0.7, we should not consider as a binding site. We should not be considered as a binding site. If the site score, druggability score is more than 1, so consider as a druggable site or the binding site. You can see, understand on what basis we can find out the binding site or binding pocket in the receptor or protein? On the basis of D score, site score. And D score and site score must be more than 1. And what is the criteria by which we are not consider if the D score or site score is less than 0.7 or 0.6? So now I am taking one examples of super oxide dismutase, or so many of you are working in the area of antioxidant. So antioxidant, what is how it can create by generating of the free radical within the body. So like O2 minus dioxygen anion is the free radical which is generate from either the oxygen or water or something. So there are some enzyme that is called as superoxide dismutase. This is called as the zinc magnesium copper catalyze enzyme, which converts what? Water, oxygen diionide into the hydrogen peroxide plus water. And hydrogen peroxide is further converted into oxygen with the presence of catalase enzyme, So our aim is here to convert the oxygen dianion, this is the free radical, into the hydrogen peroxide by superoxide dismutase, and hydrogen peroxide is further converted into the water and oxygen. So that's why free radical is just not going to, free radical is involved in for what? Cancer, inflammation, all kind of diseases or pathway, TNF alpha, interleukin, all they are been associated with that. So here, one protein PDB-ID, 2 JLP or 1 DGF. In that, there is a no co-crystal ligand is available. So how you have to dock in what area? So for that, we have to perform the site map analysis. So we, on the basis of D score, site score, I am taking only one site is here for catalase. So this is the copper zinc catalyze of the super D superoxide. So I'm not going to give the details of that. Similarly, in times of COVID, we just have to perform this SARS-CoV non-structures protein polymerase of 6-NRU. Even these, they don't have any binding site, so we have to perform the site one of this here. This is the white one, they are called as the site point. And this is the hydrogen acceptor, this is the donor, and yellow one is the, what they are called as the hydrophobic region. This is the whole proteins of SARS-CoV non-structures 12 polymerase. So I'm coming to later than first. So how many of you are in chemistry? All of you are from the chemistry background? Yes or not? So how you have to design your molecule? By hybrid designing, first of all, you go to the literature survey. If suppose if you are working on the pyrazole ring, if you are working on the pyrazolene ring, so you have to find the paper which, having the pyrazole containing EGFR activities there. So what, this is our design molecule, and this design molecule having the benzoyloxy, benzene, and this is called as the pyrazole ring, this is pyrazolene and dimethoxy, and this is the, here the, thiocarbamide. So in this, what happened, how we have to, we, from the different literature, we have to find out the most potent molecule from the literature. And in all, so in case of EGFR kinase inhibitor, one thing is very important. There are 4 types of kinase, type 1, type 2, type 3, type 4. In case of type 1 EGFR kinase inhibitor, they basically occupy the ATP binding site. So this is the ATP binding site, this is the C terminal, and this is the N terminal. And the region which connect between the C terminal and N terminal, this region is called as the hinge region. In hinge region, there is a one important amino acid that is called as the methionine 793. So which play a very crucial role for their bioactivity. So in all these, so they have to pick methionine 769, and our design molecule is also going to interact with the methionine 793. So this is the way, this is the approach by which we are going to design our molecule by searching the literatures, our own, desired molecule. Now, sorry, I have to explain these two slides already. Now, how we have to, suppose you have two compound, one is synthetic, another is called as the natural. So somebody who are working in the area of pharmaceutics, in the drug delivery or nano, so they basically combine two drug for making the nano to enhance the viability and all that. So here I am taking the, this work is under communication. So here I am taking one compound is selimarine, another is called as the epigallocatechin gallate, which is obtained from the tea extract. So how we have to perform the synergistic effect of these two? Because if we could identify one binding site for one ligand, so we are not going to bind the another ligand at the same time, because the two compound having the different affinity. So we are only, because one, those who are having the high affinity, they can bind, and the next other one is replaced. So that's why what approach we have to apply to get the synergistic by docking. So in that case, we have to identify, one is the allosteric binding site, another is called as the ATP binding site in the same EGFR protein. So here you can see this is the ATP binding site, and this one is called as the allosteric binding. So at the same time, we have to perform the dock of the two compound. They have to give you the synergistic effect. So this is the whole protein of EGFR, chain A, chain B, chain C, chain D. So sometime we see, in the literature, many who have performed their activity. So two compound can be bind at the same binding pocket at the same time. How is it possible? Because one have the affinity high, another have the low affinity. So we cannot. So there are a lot of research who has been published in the reputed journal, especially in the pharmaceutics field area. They have to superimpose both the ligand at the same binding site, and they can show the affinity. So this is the wrong practice. Did by the pharmaceutics or other person. So here, this is the epigallocatechin. This is the structures of epigallocatechins, which are represented into the stick form in the green, and this one is the turquoise color of silimarine. So you can see all these the binding residues of the binding sites. So this is the receptor view of that EGFR. Upper one is called as the, allosteric binding site, and lower in is called as the, ATP, what's called as the ATP binding sites. So you can see both the ligand is appropriately bind in a good pose. And this is the way by which we have to, how we can say that our own design ligand is actually showing the same binding pose with the co-crystal ligand present within that binding site. So for that, what we have to do? We just have to superimpose our own design ligand along with the co-crystal ligands of that particular ATP binding sites or the allosteric binding. So you can see the orientation and pose of our design ligand is same as that of the binding pose, and orientation of our design ligand is same as that of the co-crystal ligand. Similarly, in that same case of that superimposed dock pose of osimertinib, osimertinib is the co-crystal ligand, and here the ECG is called as the our own design epigallocatechin, the same methionine 793 and on that. And by molecular docking, how can you say that your molecule is ATP binding or type 1, type 2, type 3 or type 4. For type 1, there is a one called as the DGF motif. D for stand for aspartic acid, F for phenylalanine, and G for the glycine. So how can you say that your design molecule is type 1 or type 2 or ATP? So in that case, this is called as the side chain of aspartic acid in DGF motif points to the hinge region. This is called as the hinge region. Hinge region is the area which connect the C terminal and N terminal of the EGFR protein. So you can see the side chain of aspartic acid is facing toward the hinge region. So if this is facing toward the hinge region, means your DFG motif is in the in conformer, that is the type 1. And if this aspartic acid is facing away from that hinge region, in that case the DGF motif is the out conformer. Similarly, in case of side chain phenylalanine, which is away from the hinge region for the type 1, and for type 2, which is towards. So this is the way, this is the approach by which we can say our design molecule is the type 1 EGFR kinase inhibitor or ATP binding site. So before going to perform the molecular docking, we must have to first study about the whole protein structure. What are the activation loop, what is the phosphate loop, what is the alpha helix, what is the beta sheet, what is the catalytic loop, what is C lob, L lob, hinge region. We must have, and we just try to, what is the DGF motif, and we just try to dock with that particular ligand, because sometimes what happens, there are a lot of binding site. So recently few paper I have been identified where the researchers or scholars have performed the docking which is other than the binding site. So that's why earlier in case of site map analysis, first of all we must have to analyze or find out the exact binding site where actually we just have to perform the docking. So this is a site map or homology modeling if your co-crystal ligand is not available in that case. Similarly here, this is the most potent design compound 6H. So here they have to, for methionine 793, and they have to make it aspartic. So in docking there are two ways. One is called as the masterio, another is called as the glide. In case of glide, default is 2.5 angstrom, and in masterio that is 2.8 angstrom. So here I'm just within the software there is a flexibility of increase the interaction distance of between the ligand atoms and the interacting amino acid. So here 3.26 we can say this is the weak interaction, not because if your interaction is less than, distance is less than 2.8 or 2.5, you can say this is a strong interaction. But aspartic acid 1.17, this has the strong interaction is there. And we try to superimpose our adenine binding region here. Gefitinib is the co-crystal ligand of the particular EGFR. So you can see we just going to superimpose the gefitinib with that of our design molecule, and these two molecules they have the same binding orientation, same binding pocket, and same amino acid interaction. So basically in docking we have to make a hypothesis that our design molecule is the best. So if you are not going to give you a strong hypothesis, how you have to present these all in the research paper? So if your ligand is missing all these amino acid and they are binding some other area, so how can you say that your work is up to that mark? Not. So first of all, in molecular docking, you are not going to get only one PDB ID. You have to make whole, because during the COVID time, I have to generate 80 research star grids of DPP4, PPR gamma, EGFR, 80, 100. So during the data, I've just start running the system at my home, and every day I get the output. And before going to get the final result, we have to perform the dock of all that receptor grid. So how long we have to do it? Till the best results come out. So this is the way. But the student nowadays, what they do? They just giving the find out one PDB ID from the literature and dock it. So this is not up to. So you have to perform your dock of your molecule by taking more than 10 or 15 PDB ID. And before going to get the PDB ID, resolution is good. And one thing is that if your design molecule is somehow structurally similar to that of that co-crystal ligands of that particular PDB, then we have to take out that only. And this is the molecular surface view. You can see the same way as. So in research paper, how you have to represent of your docking data, this is very important. Because this is the hypothetical thing, this is the computational thing. So you have to make a beautiful table where you have to make a different column, different rows, and in each rows you have to list the list of compound, then the R group, docking score, binding free energy, so ligands of atom, amino acid, and there's the distance of interaction, pi cation, pi cation. You have to present all these data of the design molecule in this way. So this is the paper which is published in Archive der Pharmacy in 2023. Here, this is our design molecule. This is the quinoxolone compound, and here is the chloromenzine, and this one is the linker group, carbonyl NH and C double bond N. So this is our design molecule. So before going to perform the dock, what we have to do? First of all, from the literature, we have to find out what are those co-crystal, what are those reported molecule which have this fluoro or chlorophenyl ring, which have this quinoxaline ring or pyridine or something like this. So we have to perform the docking of one prototypic SB2358 bound to the P30 alpha MAP kinase. Similarly, on the right hand side, we have to perform by taking one ZL co-crystal structure. Here I am taking in the 1Y8 1YWR, and here also the pyrazolone ring. So what is the common thing with all these figures? So our molecule having the two hydrophobic moiety. One is occupied by hydrophobic pocket 1, another is by the hydrophobic pocket 2. So all I explain that all the kinase enzyme, they have the hinge region. So here hinge region, basically in case of EGFR, that was the methionine 793, or here in that hinge region, that is the methionine 109. So methionine 109, our design molecule, this is the co-crystal ligand. So we can say the orientation and binding pose of our design molecule is same as that of the co-crystals available in the literature. So this is the actual way to design our molecule. So you can see all the orientation of chlorofluorophenyl and phosphate binding site, hydrophobic 2 region 1, all that they are. So this is published in the Archive der Pharmacy. Now we have to, again we just have to find the dual pocket inhibition of sodium channel by lamotrigine. So lamotrigine is basically the anti-seizure or anti-convulsion drug which are present in just that pink stick model in the binding site of big sodium channel and the resveratrol. So nowadays resveratrol is one of the phytoconstituent. Most of the scientists who hardly left any area of activity, they just shown the Alzheimer's, Parkinson's, anti-convulsion, anti-cancer, anti-diabetic, every work, every areas of research already been found out by the resveratrol. So here I am just going to give this work is under communication. So here I am just giving the combination of resveratrol and lamotrigine, and we see the synergistic effect of these two drugs. So here we have to find out the two binding site. One is called as the site C, another is called as the site big. So in site big, lamotrigine having the high binding affinity, and in case of site C, resveratrol having the high binding affinity. So we have to dock these two drugs and the. So this is the resveratrol, here is the sodium. So basically they have to block the sodium channel. So if sodium channel is blocked, so what happened? Generation and propagation of the nerve impulse is not going to pass from one part to another part. So in case of local anesthetic or in case of so there is excessive neural firing in case of seizures or epilepsy. So how it can be overcome? By blocking the sodium channel, because sodium is one of the, calcium is one of the important cation which are responsible for generation of the exchange potential, depolarization, then partial repolarization, then of the, repolarization by sodium, potassium and all that. So here we are going to block the sodium channel by the resveratrol, and we just have to inhibit the side big B by the lamotrigine. So we can get the synergistic effect in case of epilepsy. So anti-epileptic effect is enhanced by giving these two drugs in combination. One can inhibit the sodium channel, another can inhibit the side big of. So this is the two molecules in the same binding sites. One is the lamotrigine and here is the resveratrol. They are going to interact with the different amino acids. Now, this area, this work is already under communication. So again, the same work is there. Another target we have chosen, that is called as the GABA A site, GABA binding site. Already we know GABA is what? Pentameric. They have, made up of the 5 subunit, 2 alpha, 2 gamma and beta. So this is the electron biographic of that, electron cryography. So here we can see that gamma binding site, alpha, beta, alpha 1, beta 1, and this is the gamma binding site. So this is the allosteric binding site of the GABA A. So what happened once your ligand is binding with that of the GABA A? So what happened, you can see the channel is just going to open up. So once the pore is open up, there is influx of the chloride. So once the influx of chloride ion, so what happened? That can leads to hyperpolarization, and that can hyperpolarization leads to decrease the neuronal firings, which are help in the epilepsy. So but what happened once your compound is absent in that binding site? So you can see there's the pore is closed or open? Pore is closed. So does it allow to pass the chloride? No. So there is no any hyperpolarization, no any decrease in the neuronal binding site, no any anti-epileptic activity can be achieved. So this is the way by which we are going to target the GABA A binding sites of that our resveratrol. So how you have to designing the COX-2 inhibitor? So COX-2, in case of COX-1 and COX-2, in computationally, how can you say that the COX-1 or COX-2? So there is some amino acid that is called as the isoleucine 523, and here in COX-2 that is called as the valine 23. So the molecule is big. So once we have to perform the dock at COX-1 site, so we cannot get any result of, because molecule are big and the COX binding sites, the binding sites of the bonding pockets of the COX-1 is very narrow or very small. So in that case, molecule cannot be accommodated because the isoleucine, they have the smaller and valine is the bulkier. They can allow the another bulky pocket will develop once the valence comes at 523. So here, this is the binding pockets of that. This is the hydrophobic pocket 2 and this is the hydrophobic pocket 2. This is our own design molecule, and this is called as the selectivity pocket. So here the sulfonamide group is there. So they have to bind with that isoleucine, phenylalanine, arginine, histidine, serine. This is the selectivity pocket. This one is the hydrophobic pocket 2. This one is the hydrophobic 2. So here what happened during the design of our compound, we are going to introduce one flexible benzyloxy group. If you compare this molecule with the celecoxib, this is a well-known COX-2 inhibitor, and this pink color is the structures of the celecoxib. So, and this is the green color of our stick model, that is the design molecule, that is 5U. So what happened? So this benzyloxy groups come over to that of the phenyl ring, that is the sandwich type of structure. So that's why this area, so sandwich type of structure you can see is there. So you can see this part is in the sandwich type of structure. So this is the way we have to superimpose our design molecule with that of that our. So another benzyloxy group introduced in this molecule, the purpose is that they can occupy the hydrophobic pockets of the COX-2. So how you have to design the tubulin polymerization inhibitor? So tubulin is made up of what? Alpha subunit and the beta subunit. So alpha subunit is actually designed here, represented in the purple color, and the yellow one is that of the beta chain. Beta chain, this is the alpha chain. In between, this area is called as the colchicine binding pocket. So here there's the magnesium, GTP, all that. Our compound is actually binding with that of the colchicine binding site, and we have to superimpose this compound 5 with that colchicine binding site the same way as that. So this is the way we have to design our own molecule. So this is the virtual screening we have to perform. So here what happened? So we from the data bank like zinc data bank or drug bank, we have to gather the large number of millions of compound, and through the virtual screening we have to get the model A, model B, model C, and there's the test compound, training compound, and we have to perform the 3D QSAR and hypo. What do you mean by QSAR? Any of you? Anyone can explain what is QSAR? Quantitative structure activity relationship. So what one is called as the 2D QSAR and another is called as the 3D QSAR. In 2D QSAR, we have to generate the mathematical equation between the biological activity and physiochemical parameters. In case of physiochemical parameter, what are? They are the hydrophobic, electronic and steric parameter. Steric means shape and size. Electronic means what? Electronic distribution within the molecule, like NH2 electron donating, electron releasing. How does it affect on the functional group which are interacting with the binding sites of the receptor? That is called the electronic influence. And steric means what? The shape and size. Sometimes bulky group, they are not making ideal interaction with the receptor and drug, and sometimes bulky group is also help the molecule to protect against certain degradation by beta lactamase or degradation by the MAO enzyme. So that's why it is very difficult to quantify the steric parameter, whether it is sometimes helpful, sometimes it is not bad for activity, sometimes good for activity. So it is very difficult to quantify. So this is the 2D QSAR. But in case of 3D QSAR, we have to generate the field, electrostatic field and the steric field. Okay, so suppose this is the whole room is there. So we have to divide it into a small grid or a small box is there. Each box have the different size. And now we have to identify one molecule with 3 or 4 pharmacophore is there. So we have to place that molecule within the grid box, and we have to start the grid box 0.1, 0.2, 0.3. And we have to take one probe atom that is called as the carbon, which have the plus charge. And now we have to move this plus charge carbon atom, probe atom near around the ligand's pharmacophore. And we can find out the steric effect as well as the electrostatic field effect. And then we have to tabulate it in the form of bioactive. So this is called as the 3D QSAR, that is the comparative molecular field analysis. We have to, generate the field around the molecule. Which area is good for activity, which area is bad, which is the green gene, blue region, that is actually we have to identify in that. So suppose we have taken one reference compound and 3 to 4 pharmacophore we have to put in that grid box, and after that we have taken 100 molecule, design molecule, and then we have to align all that molecule by keeping all the active pharmacophore. And now we have to taking each and every compound in that grid box, and we have to move that probe atom. I'm not doing this physically, the software did it. So they have to taking this probe atom near to that each of the atom, and we have to get the activity and field, electrostatic field or steric field. So this is called as the QSAR. So once we have to generate, then we can easily design or develop the molecule, which part we can keep in the molecule, which part which are not. So there is called as the partial least square method, PLS. So once we got is, so our molecule depends on the variables of the descriptors. So some is necessary for the activity, some is not necessary. So PLS method, what it do? It can reduce our variables, number of variable, so that what variable we have to keep? Only those variable which directly depends on the biological activity, which directly depends on the. So this is called as the partial least square method, and that giving you the one equation by which you have to get this all. So this is the 3D key as well. So we had to model C, then we have to just ligand based design drug, then design 5,000 molecule, ligand preparation, protein preparation, receptor grade, then make a virtual screening of that. Already I've explained in case of so HTBS, it require 1 to 2 seconds for, and it will take 20 seconds, and it takes the 1 to 2 minutes for each ligands to dock. And after that, we get the 5 hits. Then we have to perform the ADMET, then lead molecule, then binding energy. Then we have to perform the molecular dynamics. Then go for the in vitro and in vivo study. So this is the area, red area, what I said in 3D QSR, which part you have to keep for substituents, or which part is we are not going to add any substituent. That is all that contour map, steric contour map or electrostatic contour map. So somebody who are working in the area of natural chemistry or in that. So suppose I am taking one compound that is called as the Chicorium indicum, one plant. So from that we have to find out the 18 chemical constituents which are present in that Chicorium indicum, and we have to perform the virtual screening, and by taking that PDB ID 3W37 docket for the alpha glucosidase, 1B2I for the alpha amylase, and we get the dock score of all these. And this is the representation of that all, and here we have to get this all the data. This is also the superimposed resveratrol with that of the doxorubicin, the binding site of DNA polymerase second beta protein. So here what we see, because topoisomerase second is enzyme is responsible for the elongations of that chain. So what does T? So here what we have to dock within that topoisomerase second, they have to cleave this, they have to break down or cleave this. So by dock we can also say that our compound is cleave this chain. So there is no more chain elongation, no more making a hydrogen bonding with that aniline, guanine or thymine or cytosine. So this is the way by which we have to perform the dock of doxorubicin. So this is how it making the hydrogen bonding with the guanine, adenine, and here at the last in the beginning they have to cleave the chain. So now we also have to perform the docking studies of few molecule at the PPR gamma. So PPR gamma is just like a Y-shaped molecule. Y-shaped molecule means we have to design a molecule in such a way so our compound can accommodate with the Y shape of that molecule. So we have the arm 1, arm 2, arm 3. And there is an important thing which are available in the Schrodinger software that is called as the XP visualization. XP visualization, sometime the hydrophobicity or lipophilicity is important for what? To cross the membrane, because hydrophobicity is also required for the absorption of drug. So if your drug is hydrophobic, so it can easily cross the biological membrane and reach to the target site. Once your drug reach to the target site, what is next? Hydrophobicity is also important for drug receptor interaction according to Hans analysis or QSAR. So your drug must be hydrophobic enough to make a drug receptor interaction is there. So that's why in our molecule we have to put here the chlorophenyl group. And these CPK model, here these are the amino acid, this is a hydrophobic amino acid. So the orientation and pose of towards these hydrophobic they have. And this green part ball stick is the hydrophobic part of that molecule. So which are required for that of the cross this. Similarly here in case of naphthalene, this is the hydrophobic region, this part. And in XP visualization, we have to also showing the solvent exposed part. So So by designing or lead optimize, because one thing which are recently I just understand, that is one thing is called as the lead optimization. Once we draw our lead molecule and immediately you press, you got the dock score and interaction. If your dock score is not up to that of the co-crystal ligand, so in that case you have to remove this part and you have to add some new part is there, and you have to add some functional group into the molecule. Again you have to produce and you get the score immediately within a second. This is the, I mean, within 2 or 3 days once we start the demonstration we can. So this is the best techniques by which we can have to generate our lead molecule and compare with that of the co-crystal molecule. So our aim is here what? So our lead molecule must have the high dock score, must have the good interaction as compared to that of the co-crystal ligand. Otherwise, what's the benefit of using this software? Nothing. So if you are not going to get the desired result or output, so means this part is the solvent exhaust. So you must have to remove this part. Similarly, here in expo visualization, we saw the rotable, how many rotable bond is there. And one thing is very important in Schrödinger software, that is called as the binding pocket by surface view model. So all the DPP4, they have the S1 pocket or the S1 pocket, or here we can say that S2 pocket or extensive. So this is the binding pocket of that S2 pocket. This is the extensive pocket for enhanced activity. So this paper is published in current topic of medicinal chemistry this year. I'm just taking one of these figures. And this is the binding pocket of the surface view of model of chicoric acid. So this is the binding pocket of the alpha glucosidase. So alpha glucosidase binding pocket is very large. Sir is sitting here. Once he came by for interview of my PhD, selection committee. Actually, Professor Nasimul Huda come in Jamia Hamdard for selection committee. He asked why you have taken the alpha glucosidase for your design molecule. At that time, Alhamdulillah, I was able to answer it because our binding pocket is very large. So that's why we have to make a large molecule so that they can occupy the subsite 1 plus subsite 2 plus or minus subsite. So that's why I'm right, sir. What you asked and I said. So if your molecule is very small for alpha glucosidase, what? They have to only bind with the +1 subset. This part is not occupied. So in that case, the molecule is having the weak binding affinity. Your activity is dropped down. So that's why, first of all, we have to study the binding areas of that pocket of particular enzyme. So what are that area? What are that binding pocket? This, that. So this is also way we have to perform the binding energies of here the GABA way. So you can see these 1, 2, 3, 4, 5, 6, 7, 8 conformer is there, and they have the different binding energies. So we have taken the top one, which you can see there is the more favorable. In some part here, this molecule is out of this binding. This is the out of, and here this is also out. So out of these, which one is having the good affinity and the good binding energy is taken for the dock. So after performing the docking, what is the difference between dynamics and docking? Anyone can explain? Why we have to perform the molecular dynamics? Any idea? That can also by dock. Dock is also giving the. In molecular dynamics, we have to find out the stability of the molecule. Stability means what? So once we see the interaction between the receptor, but you never know how long this interaction can retain. How long your molecule get actually interacted with the receptor, whether 200 nanosecond or 100 nanosecond. For that, we have to perform the molecular dynamic. In case of molecular dynamic, atom level. So we have to put certain condition of the temperature, we can put certain movement, and we just allow to molecule for run. And here we have to find out the root mean square deviation, RMSD of ligand, RMSD of the protein, because we know that some part of the protein are flexible, some part of the protein are rigid in nature. So in some part of the molecules root mean square fluctuation of ligand, some of the functional group of ligand is more flexible. By lead optimization, we can replace so that it gets stable within the binding site or binding pockets of that receptor. So during the lead optimization by molecular dynamic, we can also replace our particular pharmacophore or particular part of the molecule which can be changed so that to get better stability within the binding sites. So in molecular dynamics, what we see here, so this is the this paper is actually published in March 2025. We have taken here design the Jack kinase 3. So here this one is the root mean square deviation of that of the here, that is the protein. So in case of what we have to see, can you understand this image? What you have to observe in root mean square deviation of anyone? Eh? Catches who is saying? Raise the hand. What do you? So how can you have to this time? Because these two lines are going parallel to each other. So in case of RMSD, we have taken from here. If your ligand is going beyond 3 angstrom, so we have to consider the stability between the RMSD should be root mean square deviation should be from 1 to 3 angstrom. If your RMSD going beyond 3 angstrom like 4 or 5 means so that means what? Your ligand is not stable in the protein binding site. So in dynamics we have to see the बोल तो नहीं हो रहा.
Interesting for a particular area. Interesting, and even I am learning a lot.
सबसे हमने सीखा. सर इज द मेंबर ऑफ आवर 3 PhD स्कॉलर SRC कमेटी. तो वो जाते हैं तो सर ही बताते हैं कि यहाँ पे ये करो, यहाँ पे ऐसे करो. सो आई एम स्टिल लर्निंग, आई एम स्टिल फील स्टूडेंट. जी sir. So whenever he went to Jamia Hamza, he always motivated me as well as my scholar. So they are waiting when Naseem sir is coming, when Rais sir is coming. So they are always giving the valuable inputs to our scholars and as well as the supervisor. So I can learn and impose in that our own research areas. So what I am saying, in a case of RMSD, your RMSD must be 1 to 3. If your RMSD beyond 3 means 4, 5. So means what? Ligand is losing the grip from the protein. So that means your interaction is not stable with that of the protein. So in that case, so we have to check the stability of RMSD and we have to run our dock complex in the dynamic Desmond in the Schrodinger. This is the free. Desmond is free in the Schrodinger. So we can, but for this you need to have the high graphics card and high 10 lakh or 12 lakhs of the system is there to run the dynamic systems. So here you can see these two lines are parallel. So and here 1 and 3, that is in between. So we can say our ligand is stable with that of the protein. Similarly here with that we have to compare with that of the co-crystal. How we have to compare with that co-crystal? Because whenever we have to perform the activity of our own design molecule, we always have to compare with the standard. So similarly here in dynamics, we must have to take one standard, that is the co-crystal ligands of that particular target site. And you can say the RMSD is also within 1 to 3 angstrom. So means ligand have the strong stability with that of the protein. And here this one is called as the root mean square fluctuation. Already I said that in case of protein, there the beta sheet, I mean the flexible part, rigid part is there. So these long fluctuation peaks, they can show the more flexibility in the protein in the, because we have taken a residue between 814 to 1064. This is the area where actually the molecule is actually binding. So this is the co-crystal fluctuation. And after that, here what happened, we get the 2D leak plots by dynamics. So in here, before that, one pictures I can show you. This is our design molecule, JAK3 kinase inhibitor. So here also the JAK3, leucine, and leucine 905 is the hinge region amino acid, proline, arginine, and this is the co-crystal ligand of that. Here is the leucine 900 pattern. So now this is the dock interaction, but we have to check by the dynamics whether these interaction are stable with the protein or receptor or not, JAK kinase. So for that we have to perform the dynamics. So in that case we have to see the 2D structures of leak plot. So here you can see that leucine is 905. How much interaction is there? 97%. So means here there is a strong interaction with that. Suppose I just take a bus from here and just go to a Purani Delhi, and just I'm standing and holding my hand with the roof of that bus, and during that journey the bus is moving here and there. So sometime what happened? And the duration of that journey is 1 hour by the bus from here to New Delhi or Purani Delhi. So during the journey and during the movement, what we, some part of my whole hand is not properly holding. Sometime I just lose the grip from that roof. Sometime my feet is not stable here. So during the journey, I can say I strongly hold up this hand throughout the journey, or for a second. So we can say 90% holding, we can say 30%, 40%, or 50%. So these are the point where our molecule is interacting with the binding. So similarly in dynamics, what we see, we run our molecule for 200 nanosecond or 100 nanosecond for showing their stability of that molecule. So you can see by dock, the hydrogen bonding was already observed in case of previous dock image, leucine, and there here in case of docking we have to not remove all the water molecule, but in case of dynamics we can retain all these water molecule. So these are the proline 20%, aspartic acid 11%, arginine 24, this, and this is the co-crystal also, leucine 94. So after that, there is a one fraction is there that is called as the interaction fraction. So these all are the amino acids. So we can take only interaction those above than 30%. We can take the interaction, yeah, hydrogen bonding, yeah, pi cation. If the interaction is more than 30%, we can keep all these. So here, out of these, leucine 900. So here they just the green line, they have to make a hydrogen bonding. This so the hydrogen bonding. This violet is that the hydrophobic, and this is the ionic interaction is there. So leucine 905, that is how much is around 90 to 97% is there. Similarly with that co-crystal lesion, that is the leucine is 905. So by molecular dynamics, we can perform the dynamic by atom level. How the atoms, how the amino acid is actually going to. But in case of dock, we cannot see that how much ligand is bind, 20%, 30%, or 90%. Whether your ligand is having a strong interaction with the receptor or not. So that is actually we have to check the stability by the RMSD of the ligand or by the RMSD of the protein. So, okay, thank you. So when I send our scholar, so he will giving you the demo of 3 to 4 hours here with help of the ESL. So you can understand the whole, because this is all about your passion. Docking and dynamics is nothing, because I believe that is the wet lab is very important. This is the part-time job. You can say you don't fully believe on that what pose, but this is all looks beautiful. If I'm wearing good clothes, so I look appear better. So that's called as the docking. So you cannot believe or rely only on the docking or the, but wet lab study is very important.
So no question from audience. Any question from audience? So today is second day and second lecture. Daily, you need to ask something. Yeah. Sir.
Sir, my question is like, sir,
Vikrant ji. Is that sir? You need to ask.
yes, sir. Sir, I do have a question. Like, malnutrition is one of the challenge and concern of our country. Like, the fraud marketing of like antioxidants in this food, that food. Like, I'm from Kerala and we are rich in green tea. Like tea plantations are there. And one day ago, like I have read one article, like green tea have many antioxidants. It was a biggest fraud marketing. Only
Polyphenolic compound is there, so that's
but like the hype was too high, and many of the middle class and the people who purchasing green tea, it was a little bit expensive, and but the actual studies came like this is not that much like proven, like green tea have that much effect on our like anti-cancerous, like that. Do the study and research in this area like molecular docking can do break the this fraud marketing in many food items?
Yeah, you really ask a good question. Because we have seen like quercetin, like resveratrol. These all are the, or coumarin contain. These all are the polyphenolic compound. Apigenin, epigallocatechin, because tea having the extract that is called as the ECGC, epigallocatechin gallate. This is the tea extract which have even if you go through the literature, these molecules having the antioxidant properties by in modulating the dismutase oxidase by modulating the catalase, which convert the free radical oxygen dication into hydrogen peroxide, and hydrogen peroxide is converted into water and oxygen by catalase by modulating activity. So you can, and even resveratrol, already this is the polyphenolic compound, so they can be identified good for the cancer, good for diabetes, good for Alzheimer's, Parkinson's. A lot of research work is already been done on how, and how can you say that there is no any work is that? So there's a lot of work is going on in that area. So they claim, I mean,
It have studies saying it don't have that much effect like on like anti-cancer properties. Some articles are saying like that. So this research will help, right?
Sure, definitely it can help, because there is an area one called as the nutraceuticals. In nutraceutical, they all have to perform their bioactivities of all these polyphenols, coumarin, epicatechin, gallows, catechins, and that coumarin, quercetin. They all have to be going to perform. There are a lot of area is there. So okay. So, but the thing is that in that area is a lot of adulteration, because the in the marketing they are just create a hype. So these formulation having the high of these antioxidant, these, but these the, because might be they are not going to extract the when purest form of that, and they are not going to make a formulation of purest of resveratrol, purest of all that. So that's the why you don't have to extract out the pure form yourself in the lab. So, and you can make a formulation and you can sell out. So you can, I mean, build up your own ideas to develop. I mean, you maybe one day you can be the millionaire. Okay, thank you.
Okay. Today lecture delivered by the Professor Uzair Alam is very informative, very valuable, and very learning. So thank you very much, Uzair sir. So now it is a time of felicitation. So I would like to invite Professor Kamrul Hassan sir from the Civil Engineering Department, accompanying the Nasim Loda sir. And please felicitate, and Nahid Nishad ma'am, please come and felicitate Uzair sir. Please come. Thank you. उधर से इधर ही आ जाइए निकाल के दे निकाल
सर यहाँ आ जाए ज्यादा अच्छा, फोटो ज्यादा अच्छा है।
वो निकाल के Please. Thank you. So जैसे सर ने अभी बताया we are curious do further process. I mean, sir will send his scholar to hands-on the experiment. So we will arrange the time means after lecture. जो भी हमारा tutorials में आएगा उसमें हम 1 or 2 or 3 hours का means we will arrange the slot for hands-on learning. Okay, so thank you, sir. If
And one more thing is that I will share my email ID with sir and my contact number. Whenever you design something and you need my help for designing your own way of work, so you can don't hesitate to ask me or don't hesitate to mail me. I am ready to help each of you individually, or I can assign one of my scholar. They can give you the additional demonstration and hands-on on weekend. Or weekdays or weekend basis. So you can understand. So it's all about your passion and your enthusiasm to learn. It's not possible. Everything is possible. Thank you.
so it is very good to do such a work because sir has the means uh, licensed software, not pirated. Therefore, the results will be obtained as accurate. Okay, so we will proceed this. Okay, thank you very much. So now we will go for lunch and assemble again at 2 o'clock, and then for further third lecture. Okay, bye. Thank you.